Thank you everyone for attending today's seminar, both in person and online. I will start by introducing today's speaker, Dr. Albert Braco. Dr. Bracko is originally from Germany, and then he attended the Swiss Federal Institute of Technology in Zurich for his PhD, where he was in the lab of Sebastian Jesberger, studying adult neurogenesis. He then went on to do a short postdoc with Suzanne Wegner at University Hospital in Zurich to study stroke. And I think this is where he started to get interest in cerebral blood flow. He then went on to do another postdoc in Chris Schaefer's lab at Cornell. And this is where he learned multiphoton imaging and really applied it to study vascular dysfunction, especially in the context of Alzheimer's. And so that's kind of his expertise, multiphoton imaging to study capillary stalling and vascular dysfunction in mouse models of Alzheimer's. His work has led to many excellent publications, such as in Nature of Neuroscience and Brain. He's particularly interested in the role of neutrophils in vascular dysfunction. So I invited Dr. Bracko here because, as you can see, his research interests closely align with many of ours. And I think there are many areas where we can collaborate. And his expertise in multiphoton imaging is something that has been sort of lacking at for a long time. And so it was with delight when I found out that Dr. Bracko was recruited here recently to just last year as a research assistant professor in the Department of Biology. So for those of you who are meeting with him throughout the day, I hope you'll have a fruitful discussion that can lead to many potential collaborations. And without further ado, please join me in welcoming Dr. Bracko.
Yeah, thank you for the nice introduction. I'm happy to be here today to talk about more or less my postdoc and what we have done in the last couple of months in the new lab, or it's fairly tiny at the moment. So we are a couple of undergraduates, and I try to recruit PhD students who are people who are interested in multiphoton imaging. So I changed the title a little bit to generalize it to imaging microvascular dysfunction in mouse models of neurodegeneration, to kind of reflect a little bit our recent work on neutrophils and on platelets, which are left out for today. So as you know, we can use multiphoton microscope has a big advantage over, let's say, a classical one photon, or let's call it a confocal microscope. And you can see that here on the, you can see that, wait, let's go one back. You can see that here on the picture, where you would have, if you're imaging with a laser or you kind of illuminate the whole sample, here would be your point of interest where you're imaging. So that would be your focal plane, and you bleach the sample above and below. And I hope you know that. And if you use a two-photon microscope, although this picture over dramatizes it a little bit, but you have a way less photo bleaching that allows you to image much deeper. And what does mean much deeper? With a two-photon microscope, you maybe can reach 700 to 800 microns in a brain. And brain is a highly scattering tissue. So this is the limit of a two-photon microscope. And how does it look like? So I stole this Twitter hit from a PhD student who I've trained at Cornell. So where she twittered this, where you can see what people think if I'm using a microscope and what a microscope really look like. So that was our microscope at Cornell. It's a more than one microscope. In fact, it's in fact 4 microscopes, what you can see, but it's a highly complex setup, which allows you the flexibility of imaging several organs and different angles of a brain or of any tissue as such. Now, if we use 3-photon microscope, that's images I took for a collaborative project in a mouse model of stroke. And what you can see nicely, you can fairly easy, I have to say, fairly easy. You can image up to maybe 1 millimeter into the cortex, and you can see here's the white matter. So you can see that the vessels are changing. So it's only labeled the blood vessels here. So you can see you have the layers of the cortex, you have the penetrating arterioles, and you have the white matter starting here. And if you use this non-labeling strategy, which is third harmonic generation, that allows you to image basically the myelin. What you can see in blue is the myelin, and you can see it up from the axons in layer 2 or 3, and you can see it then in the white matter, of course, where you have all the axon bundles. And a little bit more in this control mice on the right, you can see the blood vessels also nicely in the white matter, which allows you then to at least image the blood flow and also vascular damage or myelin damage in the white matter. And if you push the system further, you can go through the whole white matter as such, and you can reach the C1 region of the hippocampus, which is of course the goal, because many of the models we are working on and their cognitive impairment is somehow associated with short-term memory deficits from the hippocampus, at least in the mouse models we are using. However, that is fairly tricky because this white matter is really highly scattering and you need a lot of laser power to get through, but that is of the cost, of course, of tissue damage occurring. All right. So coming back to Alzheimer's disease, I'm sure you've seen this graph. So what I'm showing you here is from the Cure Alzheimer Fund. It's a graph where you can see that Alzheimer's disease is still one of the biggest problems for society because it still has an increase in mortality rate due to an aging population, of course. This in particular in contrast to some of the other diseases that are major diseases and causes of death, at least in the US, such as heart attack or stroke. Where you can see at least a hold or even a decline in the mortality rate over the last, maybe let's say 20 years. Now on the right, you can see a beautiful study from Ituria Medina, his now lab in Toronto, where he used public databases to study healthy patients, mild cognitive impairment, and then to late onset Alzheimer's disease and looked into biomarkers. And we only need to focus on the one here on the top, which emerges really early, and it kind of remains elevated throughout the disease progression, assuming that we have steps from mild cognitive impairment leading into Alzheimer's disease, and remains important throughout the progression. Now we looked at one particular aspect of it, which is the blood flow reduction. Now what you can see on the picture is on the left is the arterial spin labeling MRI to measure tissue perfusion in patients, which is a really nice study from Haller et al. And where he overlaid the patients. You have a normal aging patient where red is high perfusion and blue is low perfusion. And what you can immediately see if you look at the patient with Alzheimer's disease, you have a way less perfusion in the brain. And on average, we try to summarize it in a review article. It's roughly about 10 to 30% blood flow reduction, depending on the image modality or how the stage of the patient. It's not always as progressive as we thought it would be, but overall it's 10 to 30%, which is quite severe. If you look at mouse models of Alzheimer's disease, it becomes a little bit more complex. So most mouse models of disease also have blood flow reduction at a similar magnitude. However, there are also mouse models of Alzheimer's disease who does not show any blood flow reduction. This being said, so we were interested, so where's this decrease coming from? And does it somehow contribute to disease progression? So what we do as a standard technique is kind of we place these cranial windows, typically above the somatosensory cortex, just because it's a good way to reach the right matter and also catching all the layers of the cortex. And what you can see here, we place the skull with a glass window, then we let the mice recover typically for 3 to 4 weeks to avoid any surgical inflammation, which is often on the surface, and it's impairing blood flow in particular. This being said, so if you place a window and measuring blood flow, there are blood flow reductions already occurring. It's this acute phase, and if you wait a couple of weeks, it goes back to baseline. So this has been shown. So this would be a typical picture that you get, turning the microscope on, you look at it, it's a little bit tilted how the microscope was setting here. So you have the large venules, the penetrating arterioles, and the arteries itself. So we tried to measure blood flow in them, but we didn't saw any changes. Now what we did next, then we looked at the capillaries, the capillary bed, starting here. We looked at those capillaries, and there we found the phenomenon which we call stalled capillaries or obstructed capillaries. I like stalled, it kind of to imply that it's a temporary effect. It's rarely determinal, at least not in our hands. So what you can see here on the left is how such a stalled vessel look like. Now, if you think about capillaries, the diameter of a capillary, it's the same size or even smaller than any of the blood cells, except for the platelets, of course. So that means you can measure single blood flow speed in those capillaries. And that you can see here on the right, which would be a time series of a flowing vessel. You can see the black spots are moving along. Why they are black? They are black because those are non-labeled blood cells. Could be red blood cells, white blood cells, or both of them, of course. And you can see they're moving along the vessel. If you have a stored vessel, you can see that here for a given time, you don't see any blood flow, indicating that there's no capillary blood flow for this time. Now, if we look at mouse models of Alzheimer's disease, you can see here on the y-axis, it would be the fraction of vessels with stored blood flow in wild-type mice and in APPPS1 mice, which is a mouse model of amyloid beta overexpression. So it drives the APP gene, the amyloid precursor protein, under the Cy1 promoter. Now, what you can see that the number of stores are increased, but you can also see it's a fairly low number. It's maybe it's exactly 1.8% or 1.7% at any given time. So it's a temporary process. So you can imagine within the network, they're constantly around 2% of these vessels are stored. And that, of course, impacting the overall flow in the vessels. So how does it look if you look under the microscope? So this is two imaging stacks that kind of depict it. On the right, you have the, on the right, you have the wild-type mice. On the left, you have the APPPS1 mice. So stored vessels are circled in red because it's hard to see and it's a tedious process to count them. And I will talk about that a little bit later, what solutions we had to do the image analysis. Now, if we look at it, so we start on the surface of the brain and we go probably 600 micron down into the capillary bed, maybe layer 4 or 5. Okay. Okay, I see. I can do that. Okay. So in parallel, so on the right side is wild-type mice, on the left would be the APPPS1. So I would say there are two things I always like to say which are can be seen. And one of them, the overall, there are not so many capillary stores in generally, but there are clearly more in the Alzheimer's mice. So this is of note because in other models which people have now used to show capillary stalling, they typically have higher numbers than what we would see in this Alzheimer's mice, which we have 2%. And for example, if you initiate a stroke, you can have something like 40 to 60% of vessels which are temporarily stalled and then they're flowing again, or they're degenerating, depending on the model. So we were next interested, what are those black spots? So which cell type is responsible for capillary stores? So we used also the multiphoton microscope to label the blood cells. Fairly simple, it can be done with the intravenous injection of dyes. So we labeled the leukocytes or the mitochondria. It's a little bit under debate using rhodamine 6G to separate them from the platelets, which don't have the nucleus, so they don't have DNA host. You can just inject them and they label the cells. And you can see that here where you would have, okay, that's really, okay. So you can see that here, let's use the mouse. All right, you can see that here where you would have the staining. It has the nucleus and it has the leukocyte. And on the right, you can see it only has the mitochondria, meaning the rhodamine 6G staining. And on the right, you see the quantification. So roughly 60% to 70% of all the stores are white blood cells in general, and only a small fractions come from platelets, and roughly a third of them are red blood cells. So interesting for those red blood cells is, I don't know their function. They don't seem to be regulated in disease. So if we look at the stalled capillaries in wild-type mice, they're almost exclusively coming from red blood cells. However, there are other diseases like polycythemia vera, some diseases which are associated with red blood cell, or malaria, where you see a much higher percentage of red blood cells causing also those capillary stores. However, in all these neurodegenerative diseases I tested, it's mostly coming from white blood cells. So next we are asking—
Can I ask a question, please?
Yes.
Does the stall cause the reduced blood flow, or the reduced blood flow cause the stall?
All right, yes. that's already a good question. So we do think that the stall itself are causing the blood flow. We show this with modeling later on. I have the slide in still. And if we manipulate the capillary stalls, meaning we are reducing them, we see improvement in blood flow. However, that would not exclusively exclude that the antibody we are using to reduce the neutrophils could also impact other mechanisms that could drive blood flow. I mean, we cannot fully exclude it, but if we use other treatments like anti-VEGF-A or we inhibit oxidative stress, we also see improvements in blood flow. So it's definitely more than one. So we believe that the stall is directly impacting blood flow. Okay. So what are those leukocytes? So we started pretty simple with which is the most common of those, which are neutrophils. And we use this antibody here, Ly6G. So if we inject Ly6G, we realize two phenomena, and both of them are interesting. So first of all, we could show that over 90% of the capillary stalls, which are white blood cells, are in fact neutrophils. The others, we don't know. We haven't tested this yet. And they also realized if we inject a high dose of this anti-Ly6G antibody, we saw a reduction in the number of these capillary stalls. And of course, that was important for us because now it's opened up the door for us to manipulate those capillary stalls and see what they're doing in the AD mouse model, in the Alzheimer's mouse model. So first, a few words about the timing, why we renamed them. So in the literature, you can find capillary stalls as being called plugging or obstructions. So we renamed them for the reason because we think it's more or less a temporary effect. And you can see that here. So if we follow some capillary stalls over time, here only 2 minutes, but you can see when we image them in this time series, you can see it's stalled, it's flowing, it's flowing, it's stalled again. So what we think is that there are certain percentage of capillaries which have the tendency to be stalled. And I try to convince you today that there are several mechanisms that are contributing to it. And even if you look at longer time frames, we haven't quantified that as much, but you can see if we look at day 1, it's flowing at day 3, it's flowing at day 5, it's flowing at day 10, it's stalled again in day 18. So there are long-term impacts. So some capillaries have a higher tendency to be stalled. And we do think it's around 30% when we calculate that at least in this first 15 minutes, which we can easily do with a 2-photon microscope. We are limited on this longer time points like an hour or 30 minutes, just because we would get photo bleaching and we would have tissue damage, which is impacting then the flow in the vessels. So, but other people have confirmed this data using an OCT imaging, and they could show that if you go for 10 minutes or longer, you still have this roughly 30% of capillaries which tend to be stalling, even in a wild type mice. All right, so first thing we did, we injected anti-Ly6G antibody and we're measuring the number of capillary stalls. And what you can see here in this graph is again the fraction of vessels with stalled blood flow. Then you have the anti-Ly6G antibody injected intravenously into the mice, and you can see there's a reduction in the number of capillary stalls, which we do not see with a control antibody with just the IgG backbone without a targeting, so non-targeting IgG. So the next thing is we measured blood flow using the multiphoton microscope to measure with the line scan technique, where you measure the individual red blood cell speeds through a capillary or through a penetrating arteriole. We did both. So I'll show you the data for the penetrating arteriole and how we define it. We measure and we go from an artery, we go for the first branch out of an artery, and when it's branching the next time, so the second branch from an first branch artery, oh, here we go, you can see this is where we measure the points. And it's just like if you go further into the capillary bed, the flow can quickly change. And there's a lot of variation in capillary blood flow. So we try to use the penetrating arterial flow as a measurement, more of a global measurement. And we use the capillary blood flow linked to the branch number where it's coming from as a local blood flow change, which I don't think I have an example today for this. But what you can see if we measure at baseline and after Ly6G injection, you can see there's a blood flow increase, which is quite significant. It's almost a third, at least with the line scan method. So we then collaborated with Konstantin Irikola's lab and Leibach Park, where they did ALS MRI in mice for us. So it's the same pattern. So we injected, we measured the baseline, we injected Ly6G, and a few hours later, we measured the mice again for blood flow. And what you can see is the lighter colors are higher perfusion. You can see a wild type here on the left. If you inject Ly6G, we didn't really see any changes. If you look into the control Alzheimer's mice, you can see there's no change with the control antibody. But what you can see if you inject anti-Ly6G in this mice, we can see improvement in blood flow in the cortex. So we only measured the cortex. We also tried to measure using it for the hippocampus, but you can see we had quite some variation in the steeper brain regions. We just probably didn't have the resolution to do it, and with the scanner we had. So, but overall, at least in the cortex, we can see the improvement, which is the same we could show with the multiphoton microscope. So we quantified that here on the right. And what you can see is this is a wild type mice before and after. This would be the control injection and the one with Ly6G. Now we could show something which is really hard to do with a multiphoton microscope. We can show that these mice have roughly a 17% reduction in overall blood flow compared to the wild type mice. And maybe 2/3 of it we can recover using the Ly6G treatment. So just to finish this, to give you an example that this is then also impacting short-term memory, we injected anti-Ly6G and performed several measurements for short-term memory. And one of them is a Y maze. It's just fairly simple. I'm sure most of you know it. You would have a mice and it's alternating through the arms, and it will always choose the arm where it has been the furthest time before. So meaning it's alternate A, B, C, A, B, C, A, B, C, A, B, C. And a wild type mice does it at least at a higher percentage than an Alzheimer's mice. If you would have an Alzheimer's mice, it just has a higher tendency to go back to the same arm where it already has been before, which is an indicating of working memory impairment. So when we quantified that, just showing the full graph that you can see what was our thought behind it. So we did baseline cognitive testing, which would be here, those in the wild type mice and here in the Alzheimer's mice. Then we measured it a few hours later because we knew that blood flow instantly within 5 to 10 minutes is recovering. So we were also interested, is there also maybe already an impact on cognition? And then we did it 1 day later, and then we continuously treated the mice with anti-Ly6G and then did the test again, which was almost a confirmation of work which Gabriela Constantin has done in a study in 2015, where they were also manipulating leukocytes and could show cognitive improvement. And indeed, this is what we could see. So first of all, you can see there is a trend even early on for improved cognition, but already after a day, we can see these mice have improved working memory, which retains for this 4 weeks. So we can have short-term memory improvement in those mice, and we have also shown that with different tests, and we also have shown that at different ages. So we can recover it fairly early, but we're losing so if we improve blood flow in really old mice, let's say 18 months or more, we cannot recover their cognitive function with an anti-Ly6G injection. So there's a limit of what we could do, but at least until 16 months of age in those mice, we could recover it.
I just wanted to make another comment.
Yes.
We see a correlation between cognitive performance in many cognitive domains with total cerebral blood flow, both in cognitively normal humans and in early AMCI. So it's very interesting that you can see that also on your mice. So it's really amazing.
Yes, I know.
So are you aware that in humans there is a documented correlation between cognitive performance and total cerebral blood flow?
I am aware of it. There's also one, I always forget, something with gold. I forgot his lab name. There are some studies where they actively try to improve blood flow, and they also saw improvement in cognition. I don't know which surgery that it was. It was a stenosis, probably. I forgot about it. I know it's correlated, and we also have that in a review where I try to do that, but there are a few studies where they actively improved blood flow and saw improvement in cognition. So This being said, whether capillary stalls, I just take it away, exist in humans, we don't know that at this stage.
I see. Okay. Do you mind? I have to go, unfortunately. Do you mind if I contact you after the presentation for this information? I would like to include it in our paper. Okay.
Yes, of course.
Okay. Thank you so much. Sorry, I have to go. Bye bye.
Bye. So to summarize all this previous work, which you can read up, it's like, so I try to depict it that you can see we also took some other measurements, but because of course we wanted to know where what is causing those stalls. And first we really looked into amyloid beta, and we couldn't really correlate it to amyloid beta. However, what we did saw is, so if we have the stalled capillary, mostly caused by white blood cells, in fact by neutrophils. So if we give anti-Ly6G, we saw improved blood flow and we saw improved cognition. We saw amyloid beta burden as a reduction. So I put the arrow down, but in fact, we only saw a change in A42. And I spent a lot of time trying to work, what could this mean? Since it's more associated with blood vessels, but I just couldn't, or A40, I'm sorry, A40, and I couldn't really narrow it down. I never could find the mechanisms where you could show, are there more cerebral amyloid angiopathy plaques or anything like it? That didn't work out. So I never could really correlate it to plaques until today. So if we have then a control, we still have those stalled capillaries and we have the reduced blood flow. And this is of course temporary. So one injection, since leukocytes have or neutrophils have a high turnover. So with time, the cognitive impairment comes back, of course. So how do we measure all of this? So we created this online game, and it's called Stalled Catcher. It's a run company now, which is driven by Pedro Micolucci. He runs, okay, here we go. He runs the Human Computation Institute. And he helped us to set up this project. So what people do, so people get those stalled capillaries. I just have to say, we really tried hard to get an AI code or to get an machine learning code that counts those capillary stalls, because we had many undergraduate students who counted those stalls, including me, and it takes a long time for each stack to really find them because there are only so few. So we developed this game where people need to make a decision whether it's stalled or flowing. So if we look at it, this is how the layout is of the game. That's my account. As you can see, I never really counted anything. But what you can see is that people can scroll through here and they get a capillary which they have to look for, which is inserted in red, which we can do with a computer. That's a MATLAB code. And what you can see is there are over 45,000 players now. Of course, a lot of them are not playing, but there are certain players who spend, we know what each of them is doing, several hours per day playing. And this is what they do. And I even met a couple of them, so it's really successful on 2 levels. The data analysis is tough because there are quite some errors in still as well. But also from an advertisement perspective, it's just really nice to engage also with the community, with Alzheimer patients and families with Alzheimer's disease. So it has been a really good project. So really successful to working out. So this is kind of a look into my brain for Alzheimer's disease and blood flow reduction, and where do I think it's coming from. As you can see, the arrows are all going back and forth, so I'm a little bit uncertain what I can say. So, but we definitely have centralized the blood flow reduction that's definitely interacting with Alzheimer's disease. So whether it's vice versa, I'm not sure. What I can say, so aging drives blood flow reduction itself, which is really hard in the mice to tease out. But if you have an Alzheimer mice that overexpress amyloid beta or some tau models, not all of them in particular, you can see also blood flow reduction. So somehow there has to be a correlation from amyloid beta to blood flow reduction. We just really haven't found it yet. So if you look at the left, I would say those are the hits or cardiovascular risk factors that interacting with the blood flow. All of them are associated with blood flow reductions. I mean, some, you know, it's TBI or stroke is easy. So I was looking into exercise and obesity. And although those models had blood flow reductions, they are really not interacting with those capillary stalls in the microvasculature. So we published those, and which I will not talk today, is this we just recently published a month ago, and this study where we looked at the blood-brain barrier leakage. So I left it out for today because it doesn't have as much of the nice 2-photon imaging, which I'm trying to show you today from this unpublished data. However, what we're showing here that if you use an anti-VEGF-A antibody, you can reduce the permeability of the blood-brain barrier again, and this also reducing the number of capillary stores, and it's also impacting blood flow. Now, if you look here on top, if we thinking about what could also impact the blood flow, we were thinking about oxidative stress and vascular inflammation. So we sought out a couple of years ago to image that using in vivo imaging. Imaging inflammation in the vessels, it's not so easy if you look for the receptors, and in the capillaries, it's in particular difficult. So it took us a long time to get any data, just because the labeling and to make sure that you specifically labeling the inflammatory receptors and not the plasma or unspecific binding. It took us a long time, but I think we're on the right track, as you will see in the next couple of slides. We're trying to convince you that oxidative stress is also impacting capillary stalling and blood flow. Now, if you look at the hypothesis we have by now from us and also from many other groups who have picked up those capillary stalls and are also working on them. This is not a full list. This is mostly a list for the first who have done it in a certain model. So it's not appreciating all the people's work. But if we would have a flowing capillary, I showed you the store rates can between 2% to maybe 40 to 60% if you have a TBI hit. For example, it's the most massive insult you can get. And there are several theories out there which I will not talk today, but we have shown it's involved in the blood-brain barrier. And other people like Andy Shee's lab in Seattle or David Atwell, they have shown that there's pericyte constriction, which may also impact capillary stores. The direct correlation of capillary stores to constriction is still lacking. It's not as easy as a measurement to measure really the diameter and precisely in the small capillaries. It really takes to the limit of the microscope what you can do, at least in vivo. However, they have shown it also with human tissue. So it's quite beautiful work. And what I've shown you today is going to be, we're talking about vascular inflammation. And then I just want to point out again, so most of them are only there for a second or maybe 5 seconds or 10 seconds up to a minute. But there's one study who showed that they can be 20 minutes and longer. And this is work here by Patrick Reason in Craig Brown's lab. So they have image stores for 20 minutes and longer. They specifically looked only for those. And then they could show, which we haven't seen in our mice, is they could show if they're 20 minutes or longer stalled, they tend to get degenerated. So you see string vessel formation. So it could well be that the capillary stalls over time are also degenerating those capillaries. And although the data is also not as clear for capillary degeneration in Alzheimer's disease, I think it's safe to say that it's occurring. All right, so do capillary stores exist in humans? So we try to address that with modeling. So this is microvascular modeling done by Maxime Berg. At that time was a PhD student with Sylvie Lautroy. She's in Toulouse and she's a modeling person. And what she did, she took the data set from David Kleinfeld's lab and introduced into this mouse cortical section capillary stores, similar to what we can see in vivo. So it's the same percentage. And on the graph, you can see they're increasing the percentage of stores. And then they looked into human data. So this is a human vascular network. They traced the network, defines the vessels, and you can see it's fairly similar, but the capillary network is not the same between humans and mice. So there's some variation. However, when they introduced capillary stores, they could see the same magnitude of blood flow reduction, what we could see in mice. So is there anything else out there? All I can say, if you look at diabetic retinopathy patients, I found this older review from Lee from 2007, where they have this autoradiographies where you can see here capillaries. And it's known that you have blocked capillaries in the disease. So you can see nicely here, this looks exactly as our 2-photon imaging. You can also nicely see it here. So blood vessels are labeled. And then interestingly, if you look at those arrows, what is labeled in those vessels, so these are in fact leukocytes. So it looks like that there are also leukocytes that block those vessels. However, you cannot exclude here that these are just post-mortem leukocytes that are got stuck in the blood vessels. So although although that try to be perfused, but of course you cannot exclude it. But you can see definitely stalls. There's also reduced blood flow in the disease, and there's also a strong inflammatory component. So to kind of what we could do, thinking about it, we could use a diabetic model and image the flow in the eye. This maybe could be also done in patients using OCT imaging, where you're close to the resolution of imaging capillaries. But I think in a few years from now, we probably have that answer. And if you think about it, for the disease, as a treatment, you can use anti-VEGF-A antibody, which we also use to reduce capillary stalls. So there's a nice link for the future that maybe could help to kind of prove that stalls exist in Alzheimer patients. And then we could go from there and thinking about, could we maybe improve blood flow in patients, and would that be beneficial? I think that remains to be seen, whether this can be transferred. Now, the last few slides, or a couple of slides, I want to talk about oxidative stress, that plays an important role in Alzheimer's disease. And it's particularly critical for the vasculature, where you have the production of reactive oxygen species, which cause then damage on the tissue, in particular on the membranes. And this leads to increased inflammation, and this may recruit leukocytes, and as such then cause capillary stalls. So if you look at this modified picture, you can see the idea would be we have blood flow, and we have the neutrophils that kind of bind to this ICAM, so the CAMs, which are the inflammatory receptors. At least in that case, it's a capillary, or I want it to be a capillary, where you would have the receptors. And you can imagine if you have more inflammatory receptors, you have more probability of the leukocytes to get stalled. And this is then even accelerated in Alzheimer's disease, because there's a vicious cycle of oxidative stress and amyloid beta, which somehow cause tissue damage. So then with our collaborator, which is again, Konstantin Iadicola. So we worked out on his pathway, because he has an oxidative stress inhibitor that he's used commonly in his lab. And you may know is NOX2. That's a membrane-bound enzyme which is converting the NADPH-dependent oxidase. And we have inhibitors to use them. We can block it, and as such, we can reduce the oxidative stress in the tissue. And in turn, it leads to influx of calcium, and we think it also leads to increase of vascular inflammatory receptors not being on the picture. Interestingly, the complex itself can be activated by this receptor CD36. And you may know it's a scavenger receptor. It can bind A beta itself, but it can also bind all the pro-inflammatory cytokines like interleukin-6 or TNF alpha. They can also bind to the receptor and then drives this pathway. So we did that together with these two students, which was one of my first undergraduate students. She worked for me for 3.5 years in Cornell. It's now a medical student in Yale. And Nancy Ruiz, who took on the project later on, was a PhD student, or is still a PhD student for 1 more month at Cornell, and then she's graduating. So we seeked out, kind of try to repeat our data. And what you can see on top is the mice injected with an inhibitor. It's called GP91. That's a NOX2 specific inhibitor. And we have a control peptide, which is the scrambled version of the same inhibitor. And what you can see, so if you use the scrambled version, you see increased number of capillary stalls here in yellow. And if you inject the inhibitor, you see a reduction in the number of capillary stalls. So this is the same mice. So this is paired statistics, because we can image the mice before and after we inject it. That's easy. And this kind of repeating what we have seen before, we basically repeated our own data to show that most of them are leukocytes, but there are also different forms of stalls as caused by platelets or leukocytes or by red blood cells, as I said. So when we quantify that, you can see here's the stall rate. And it's a big graph. You would have here the wild type mice, there's reduced stalls. You have the APP mice, there's increased levels of stalls. And if you look at the Alzheimer mice, although it's a little more variation, of course, so after 1 week, you can see there's a reduction in the number of capillary stalls. And this reduction in number of capillary stalls is also impacting blood flow, similar if you want to Ly6G, but on a really different time scale. So Ly6G we can inject, and after 10 minutes we have improved blood flow. If we inhibit NOX2, at least with the small peptide inhibitor, it takes us at least a week before we see increased blood flow again. And you can see that here, that would be the volumetric blood flow. So it's the flow speed and the diameter taking into consideration. You can see there's an improve in blood flow. There's also an improve in speed. Of course, they are dependent on each other. And we really don't see much of constriction happening in those vessels. So if you look at the diameter, it remains fairly constant. Of course, we're not measuring as sites of parasites because at that time we didn't have the parasite reporter in our mice. So we cannot again exclude parasite constriction contributing to it, but at least as of now, we also cannot prove or disprove it. So here we tried to look into inflammation. So we did a fairly basic analysis of activated microglia using IBA1 and also in astrocytes using GFAP. So it's a little bit like what we see for amyloid beta. So the changes are somehow there, but they're not significant. So if you look here, for example, just focus on the IBA1 staining. In that case, it would be the red cells. Green would be GFAP, and the plaques are stained for in vivo staining of the plaques, where we use methoxyXO4, which you can inject into the mice, and it stains any protein aggregates in vivo. It's a really nice dye. It's a little toxic. One really needs to take care of the amount you inject. So, but what you can see, there's a trend of if you have the NOX2 inhibitor in Alzheimer's mice and in control mice to be reduced in the cortex, no change in the hippocampus. So we are not sure what it does on inflammation on the overall tissue. And the same you can see for GFAP, there's a little trend to be less, also in the cortex, but not in the hippocampus. And if we look at amyloid beta plaques, it's kind of the opposite. So I really show you how the data looks like. If you look at cortex, you can see there's this huge variation, and this is not an outlier. It's still within the range. No change in cortex, but there seems to be a reduction in the hippocampus. So we weren't really sure what we should make out of this and what we should draw for conclusion. So we decided looking into vascular specific labeling. So we try to label the CAM receptors. So, and we did this in this kind of scheme where we have placed cranial windows. We waited for almost 4 weeks. Then we started to treat the mice with a NOX2 inhibitor, just because we wanted to see whether we can get changes upon reduction of oxidative stress. And then we injected intravenously the receptor. So we had in VCAM, we had an P-selectin, and we had an ICAM, and we had a PCAM, together always with a vascular staining. And this was quite tricky to establish the staining because in particular, it's hard to see it in capillaries. It's easy to see it, as you can see, for example, here on an artery. You can see anti-VCAM staining, and you can see it's nicely lining the vessel walls, and the plasma label would be Texas red labeled plasma. If you use a control antibody, you really don't see left. So really find good time points where only specific label remains, which is typically between 12 hours and maybe 1 day, depending on the combination of antibodies we inject. Now, this being said, so we did that, and this is kind of a sneak preview into this data. These are Alzheimer's mice, but you will see the quantification on the next slide. So this is not a done figure. So this is really like how it looks like before we really process the images. So this is fairly raw data, I would say, which I think it's good because you really have to trust your staining here because it all depends on the quality of the staining, whether you can really quantify the data. And it's really hard to quantify the fluorescence intensity in a multiphoton stack, whereas a non-linear noise that is going down with depth of the images. So, but overall, we can make the stainings work. And you can see that, for example, if you look at VCAM here in a penetrating arteriole, you can see we nicely see the membrane staining, or if we take a transversal section here, you can see it here. If we look at the capillaries, you can see it, it's there. It's fairly fuzzy. It doesn't look as membrane, but it's really hard to the diameter to get the resolution that it would look as nicely only on the membrane. But we were quite happy to see it. And for example, if you look here at ICAM, in a venule, for example, it's really nice in a venule. It's induced, so that's an Alzheimer's mice. If you look in a capillary, we can also see it. If you look at P-selectin, you can see it's hardly any P-selectin we see in the capillaries. If you look at PCAM, you can also see it in the capillaries, and that would be the labeling of the blood vessel. One is Cascade Blue and Texas Red. They're both labeling the plasma. So that really worked nicely. So we can label them. So we try to quantify them. Just go one slide back where you could see we really try to choose region of interest and try to use always the staining of the plasma label as a background comparison. So at each step, we try to compare the fluorescence signaling of the antibody we injected to the intensity of the plasma labeling, because we believe at least the plasma label should reduce in intensity the same as the antibody labeling, which we can assume, but of course we don't have a real proof for that at this stage. So if we look at this data where we looked into it, so we would have the graph here for penetrating arterioles, which I think is definitely promising to work on it. And you can see this would be the Alzheimer's mice with a control scrambled peptide. You can see we see increased levels of the stainings in the vessels, similar what you have seen on the pictures. If we inhibit it with NOX2, you can see a reduction in all of these in all of these penetrating arterioles. Let's say it's this way for all combinations, which would be VCAM, PCAM, ICAM, and P-selectin. Now, if you look at capillaries, you can see the picture already is different. We could only see an increase in VCAM, which then is also mitigated in the APP with the NOX2 inhibitor. And this is really interesting because there's quite a push for anti-VCAM antibodies in Alzheimer's disease driven by a couple of labs. So maybe it also has an impact in the capillaries. I have to say, when you use single cell sequencing of the capillaries, you don't see a difference, at least in the one group who has done it, between Alzheimer's and patients. There could be many reasons for it. I think if you try to extract the capillaries out of the tissue, you may already increase inflammation. So I'm not sure how this data looks like, but I just want to point it out. We definitely see a clear increase in VCAM in the capillaries. All right, so we try to also do that staining post-mortem. For just one example of these, which is the VCAM, we focused on VCAM because it has the strongest impact on the capillaries. So we try to stain it. And you can see the staining definitely works. If you look at wild type, you almost don't have any VCAM staining. It's really fuzzy. If you look at Alzheimer's mice, treated or non-treated, you can see there is staining in the capillaries and in all of the vessels. If we counter stain it here with PCAM, for example, and we quantify it, you can see there's in the wild type, there's basically no staining. So we normalize it to 1. You can see there's a reduction, and it's just not there yet. We just have to count more animals. Maybe it is also reduced. It would at least strengthen our in vivo data, but we just have to see. All those ICAM stainings or VCAM stainings are really difficult in tissue section. So all of them took us a long time to try to establish them with the controls and reliable stainings of these, unfortunately. So we also did it on human tissue, and we hope to do it with immunofluorescence, but we had some problems to make that work. I don't know whether it was our tissue from the brain bank or our skills with human tissue. This has been done before, so it's known that NOX2 is increased in Alzheimer patients, and we also do see that, but you can see it's not the best staining yet. But you can see it if you, so you basically would not expect it to see it much in control patients in any case. But it's hard to see it on some vessels as here. But if you then look in Alzheimer patients, you can see it on the vessels and also on the neurons. So NOX2 is also on all of the neurons. So you can also see those also being labeled, and we try to quantify that. So NOX2 is definitely increased in Alzheimer's disease. We hope to counter stain it with lectin on the vessels, but we just had problems to get the staining going, unfortunately. Yes. So what I'm trying to show you is that we have demonstrated that NOX2 is important for capillary stalling, for blood flow, and also for cognition, which are left out, and also for vascular inflammation. So NOX2 also is rescuing cognitive function, which also has been shown before, and we also do see that in our mouse model. And then we have VCAM as a potential candidate to be regulated by, or at least impacted by, oxidative stress, specifically in the capillaries, but also in other vessels. But we focus on the capillaries. And then now we also have RNA-seq data. We just obtained it. I cannot say much what is going on, but our idea was to extract only the capillaries and try to run RNA sequencing. So why do we think we can maybe make a difference than the other groups? Is we do believe that we found a protocol that is really minimal in increasing inflammation, and we at least could show that. So if we look at inflammation markers in the wild type mice and in the Alzheimer's mice, we can see differences in the capillary fraction we try to extract. So this could be promising, but I think it remains to be seen. We have the data, but we just haven't analyzed it. Now to put that also together, because it could be well that all the data we've seen kind of plays together. And as always, it could well be, and it's probably likely, that capillary stalling contribution to blood flow reduction is always a secondary effect generally that occurs upon inflammation likely, or upon something like stiffness or changing in mechanosensory receptors. Now, we could show that NOX2 is involved in it. I showed that today. We could show that VGFA is also impacting the number of capillary stalls. And those are the two mechanisms we have teased out today. And then also other people have shown that pericytes are involved in it. So for me, like, I became interested in why not look at the neutrophils themselves? Because there's some nice data, which I will show you from other groups for a slide or two, that show that there are different fractions of neutrophils, and some of them are regenerative and others of them are causing disease. So, and this is a summarized, somebody made the summarized picture of this paper that came out in 2020, as you will see next. So this one was then in 2021, which was just a summary, where they did single cell sequencing of those neutrophils, which they got from mice before and after a nerve crush. And we look at the data. So you have all the maturation stage of a neutrophil. And if we look at the data, I just want to highlight here their baseline neutrophil phenotype. So here the yellow or the orange ones are immature neutrophils, and the amount of them they're caught. Then you have this intermediate neutrophils, and you have the mature, more like probably disease-causing neutrophils here in green. And if you then look into the mice with a nerve crush, which will here on the right, you see a strong shift of the neutrophils. So you have now much more mature neutrophils. You have less intermediate, and you have less immature neutrophils. So what does it mean? They did not look into the damaging causing neutrophils, so they were interested in regeneration. So, oh yeah, I forgot this one. That's also important. Just pointed out, because one of the genes they found to be separating the two maturation stages of neutrophils, or the two subpopulations of neutrophils, was Ly6G. And Ly6G is also the gene we were working on. So maybe Ly6G itself already defines a disease-associated neutrophil, but this remains to be seen. We don't know that in Alzheimer's disease. So what they did, they looked into axonal regeneration, if you want, and what they did, they kind of sorted out the different populations, and what they could show that they have more regeneration if they use the regeneration fraction only of the neutrophils. So this is quite promising. Of course, my idea would be, is there a subfraction of neutrophils that driving the capillary stalling? I don't have the data yet, but I have some preliminary data to convince you that neutrophils are different in Alzheimer's disease. So the idea would be having Alzheimer's mice, having wild type mice, extract the blood, do single cell sequencing, and plot the data. And then, of course, at a later stage, it would be nice to do in vivo imaging of the subpopulations. And then we would functionally test whether we can find also regeneration fraction in our assay. So what we do, or what I've established is, for this one, is using adoptive transfer of neutrophils. So we take the peripheral blood and we FACS sort for neutrophils, and then I label the neutrophils. We could use the transgenic mice, but the idea would be to use an external label that then could also potentially label those subpopulations if they exist. And then we kind of put them into wild type mice and take the output measurements we can easily do, like neutrophil extracellular traps, vascular inflammation, blood flow, of course, and capillary stalling, and pericyte constriction. So we can do all in one mice, or in one group of mice. So I have some data on that to convince you that we can, that neutrophils from Alzheimer's mice are different. So what you can see here, If we do adoptive transfer on those mice, and we label the neutrophils, in that case here with a Ly6G, because I just had it in hand, and you put them into wild-type mice, you see capillary stalls, and you see an increased number of them. So to cut this one short, this slide is, if you transfer Alzheimer's mice from 9-month-old mice, so quite late into the disease, and put them into wild-type mice at the same age, you see an increase in capillary stalls. If you use wild-type mice from the same age and transfer them into wild-type mice at 9 months, you don't see that. You see that still the number of capillary stalls is decreased. So similar to a wild-type mice. If you do that in young mice, you can see it's a little bit all over the place. So if we have wild-type mice and use an old neutrophils and put them into young wild-type mice, you see a trend for more capillary stalls, but it's not significantly different. Also with the mouse numbers I had here. And if you use Alzheimer's mice that are young, you see, of course, more stalls in the Alzheimer's mice, but you don't really see a difference if you use a neutrophil mice from an older mice and put it into a younger Alzheimer's mice. So it looks like early on, we don't see a difference, but if you look at this 9 months old time point, of course, we don't know what is happening in between, but there we see a strong effect on those neutrophils. So somehow the neutrophils become more maybe activated or hyperactivated would be a good word I would like to use for those. So hyper or reactive. So they definitely change and they cause damage in the microvasculature, at least on the level of capillary stalling. If you look at the blood flow, it's complicated. So I couldn't do the penetrating arteriole, and the students need to analyze the data. But if you look at capillary flow, you can see there's not really a difference yet from the ones I measured here. So you can see 9 months got the neutrophils from 9 months old and the wild type from wild type. So it looks like there could be something, but as of now, there's just no difference yet. So we have to see or wait longer or maybe deplete the neutrophils beforehand. So we just have to see what we do here to maybe tease that out. Or it's not strong enough to drive it. I think it just remains to be seen. So the idea is then, of course, maybe we can also do that from patients. So could we use patient neutrophils from Alzheimer patients and adoptive transfer them into the mice? That definitely sounds cool, but it has its problems since sizes of neutrophils and capillary diameters are not one-to-one translatable. So maybe just human neutrophils are causing capillary stalls on a baseline level. However, it's possible to do, and of course would be a nice thing which we could also do here in Miami. And then, of course, to see what damage they do in vivo, and if we can label specific subpopulations. This kind of the process for doing that has been done for CAR T cells, really popular, and many clinical trials are using it. And they also have their working line of where they use the cells, take them into cell culture, but then image them in mice, and then later you transfer them in patients. And maybe this would be also, of course, a path forward. I'm not saying into patients yet, so I kind of restrict myself to this part as of now, where we could use human cells and try to image them then under the multiphoton microscope, which is in theory easy to do, but we just have to see how the differences between mice and neutrophils and human neutrophils play out. To finish this, I found this one also from Miami, where people have used the peripheral blood and kind of isolated it and worked on it. So I really think this could be something doable with the people here to make that happen. I think that would be a really cool experiment. And then we see what those neutrophils could do and if they're also driving disease. So what I've shown you, their capillary stalls contribute to blood flow reductions. I think that's pretty clear. And then capillary stalls likely caused or at least contributed. Vascular inflammation is definitely contributing to capillary stalls and not vice versa. Maybe also vice versa. We cannot exclude that. There's inhibiting reduced CBF and the associated reduced number of capillary stalls. So it's a correlation, of course. We don't have a direct mechanism. So what I didn't show you, we have blood-brain barrier leakage that also contributes to the phenomenon. And then that disease-associated neutrophil exist, whether those are subpopulations or the whole population of neutrophils, of course, could be different, which would be a little less exciting, I would say, but it's still, of course, worth looking into it, because I think neutrophils have been overlooked in the past, and they're clearly contributing to chronic or diseases such as Alzheimer's disease, in particular in the vasculature, and as such also in the microvasculature. And with that, yeah, I just want to thank the lab. So most of these experiments I did myself still at Cornell because we don't have a running microscope yet. There's supply chain issues with optical parts, unfortunately, to kind of construct the microscope at the moment. So we only get bits and pieces, and we are missing important pieces that we need to really make the system going. So I have this few students who are helping me, and they're really good computational undergraduate students here at and they do a lot of their image analysis, like the capillary stalling, the vascular inflammation, and the data you're seeing. It's done by these really excellent students. And the collaborators, which I unfortunately couldn't talk about, but I want to point out that I'm also really interested into platelets. If somebody of you is also interested into platelets, so we have a way of imaging platelets, and I myself are particularly interested to look into platelets that directly binds to neutrophils and maybe drive damage once they're interacting with each other, which probably is true, and we try to image that in vivo in the mice. And then with Miguel Perez here, we're working on a vascular dementia mouse model and looking into blood flow and other mechanisms, and of course also Cornell collaborators and Cornell University from my postdoc time, which most of the data came from today. Thank you.
Okay, so we're a little bit over time, so maybe we can take a couple of quick questions.
Our Zoom listeners, if you have any questions, just go ahead and unmute yourself and shoot.
There is a question on Zoom.
Can you please read it, Maria?
Yes, hang on just one sec. What is the proposed mechanism by which LYS6G inhibits neutrophils from stalling flow?
Yeah, I mean, so I jumped over that just because I think it's really complicated. So with time, Ly6G is depleting neutrophils. So this is really well established. In fact, Ly6G is used to deplete neutrophils. We do think there are two effects in play, which we haven't really teased out at the moment. We do think we have an acute phase of Ly6G, where we've shown by flow cytometry that there's no depletion occurring in the first few hours after injection. So we do think we have here an inflammatory component, which we're not 100% sure how to explain it at the time. But what we know is that the blood flow comes up immediately and the stalls are gone immediately when we inject it. So with time, if you look a day later or 2 days later, the neutrophils are depleted. So I think we have two mechanisms. So one of them is depleting. So you take the damaging causing neutrophils out and you see improvement. But we do think there's also an early component where we don't know the mechanism yet. Now, I'm not sure we have to look for that at this stage, but it's hard for me to speculate even what could be going on so early.
Okay, other questions? All right.
Hi, thank you so much for your talk. I just have a quick clarifying question. I apologize if you mentioned this already, but is Ly6G itself interacting with endothelial cell receptors like the ICAM and VCAM, or are these two different, parallel phenomena?
We do think it's parallel phenomenon. So the function of Ly6G is not well established. There's one paper from 2012 coming from Rochester, I think Wang et al. In blood. It's definitely in blood, and where they show it is interfering with integrins. Your answer could be yes. We tried to inhibit the integrin itself, where it supposedly binds. We didn't see a difference, but I have to say there could be a messy experiment. I'm not sure that rules it out. So yes, it could be, and that could explain this early phenomenon that we are also seeing.
Yes, welcome to
We have
Miami.
one other question.
Welcome to Miami, and great to hear this beautiful talk. Have you looked at vascular reactivity in terms of some of the arterial data that you had? I mean, that's a real important area in terms of blood pressure regulation and things like that. And many times in the Alzheimer's patients, you're having fluctuations in blood pressure. And I just wonder if you have any data on vascular reactivity?
So we haven't done vascular reactivity on these. This is an ongoing project in my previous lab. So they're trying to do that in awake mice. I haven't done that myself. I cannot even give the data. It's not a secret, the data, but I think they have a harder time to get this in awake animals done. So it would be, you could do a viscous stimulation, of course. That's what they're trying to do. And I don't know, it's kind of established, but they have trouble. That's all I can say. So we have to see how this turns out. Yes, it's definitely impacting it. What I can say, that hypertension itself is impacting it. So I have implanted pumps into mice with angiotensin, and that's impacting the capillary stalls and the flow as well. So that I can say, but we haven't really done the functional measurements. People have done it in wild type mice. So saying this, there are changes.
Mary, was there one last question from Zoom?
Yes, one additional question. Did you examine changes in parasite coverage, morphology of stalled versus flowing capillaries?
Yeah, so I would love to do that as well. That's a really excellent question. So the coverage are reducing in those Alzheimer's mice of parasite, but I have to say, so we have so since it's a few capillaries that are stalled, it's really, really hard to find those capillaries again once we sacrifice those mice. It's just so few, and you really have to go through the map and find them. We have done that in our recent publication, where we have that in a figure where we did that, but in reality, we found 7 or 8 stalls again. We didn't look where we then looked for tight junction proteins and saw changes in vessels that are stalled or that are not stalled. So we do think that the capillaries that tend to be stalled are different, but we haven't convincingly shown that yet.
Okay, because lack of time, we'll just wrap it up here. Thanks again, Oliver, and look forward to the rest of your day. Thank you.